We raised back in May, but we decided to keep things a little bit more quiet until we had something to be proud of, which is not to us raising money, but actually delivering something that makes an impact. After we left New York in January, the first place we went was San Diego, where we started working with our first lab. We recently delivered to that lab like 2 weeks ago, November 11th, however long ago that was. Thank you. But they're a group that had an interesting problem. It was a rare disease gene therapeutics group. They developed these RNA therapeutics called antisense oligonucleotides, which is just like too fancy of a word to say these short sequences of RNA that are able to go into the cell and find mutated mRNA and either bind to it right there and block it from getting translated in the ribosome or digest it. So it's really cool. It finds the bad stuff, and it's like a little genetic missile that goes in, finds the bad mRNA, destroys it. So these therapeutics can be used to target a variety of different diseases with a genetic basis, like muscular dystrophy or Huntington's. Really cool stuff, but the issue is that for that genetic missile to hit its target, it first needs to get into the cell, and that's a tough process. So researchers attach something called conjugate, which is like a molecular key that binds to a receptor and allows the missile to go in. But every single cell type is going to have a different set of locks and keys that work with those locks. So to find these conjugates, you have to do a lot of experiments. It's like looking for a needle in a haystack. To date, there's only one conjugate that's approved by the FDA, GalNAc. We have over 200 cell types in our body, and we've only got one key for one cell type. We're restricted. So at La Jolla Labs, they had the idea to use automation to look for new keys. They paid some integrator north of $1 million to set up a work cell that was capable of sifting through all of these potential conjugates, running these experiments, iterating on them, choosing a new conjugate to find these needles in the haystack. But what they discovered is what, unfortunately, a lot of people discover when they try to adopt automation, which is it's not made for this type of thing. It's not made for high differentiation work. There's no cognitive abilities of these work cells. You have to program them in a very meticulous, rule-based way. So the program failed. They still have to rely on human labor, these really, really sharp researchers, to go and do a bajillion RNA extractions and qPCR plate preps. They're bottlenecked. So the interest to us was creating a tool that helps them find these needles in a haystack, or at least help them with part of their workflow, so they can rely less on their physical labor and more on automated physical labor to do this pretty hard task and solve a cool problem.